Hi, everyone. Just want to thank you all again for coming and participating, and thank you again, Dr. Miller, for a really outstanding talk. My name is Paul Barrett. I am a member of the Office of Strategic Coordination, otherwise known as the Common Fund at the NIH, and I'm very excited to be here moderating the first session for the Sex as a Biological Variable workshop. We'll be focusing in this first session on sex differences in brain function and behavior. We have some really great talks lined up for you, but I also want to encourage everyone to attend the poster session this afternoon from 3:30 to 5:30, because we have a lot of great posters that will also touch on this subject. So I think everyone here can agree that sometimes there may be a slight difference between how men and women behave and think. Despite these apparent differences, we're really only starting to scratch the surface of understanding the biological mechanisms that lead to these variances. Researchers have shown that during development, sex may contribute to cognitive functions such as learning and memory, fear, or language development. For example, neurological disorders such as dyslexia and stuttering are 3 to 4 more times prevalent in males compared to females. Conversely, women are about 2 times more likely to be diagnosed with major depressive disorder when compared to men. In addition, sex has been linked to the age of onset differences in different neurodegenerative diseases such as Alzheimer's and Parkinson's disease, as well as one's ability to recover from traumatic brain injury or stroke. Despite all of these differences that we've noticed, males remain the research subject of choice in neuroscience, with roughly 5 times more studies being conducted solely on male animals. This is just one example of the type of trends that the SABV policy is trying or aiming to amend. When considering the impact that sex has on neurological development, function, and health, one's first instinct is to consider maybe how external factors within the body can influence the brain. For example, we know that during puberty, males and females release different hormones at different levels, and these can alter different brain functions as well as the architecture of the brain in different regions. Other external influences, such as one's gut microbiome or one's metabolic profile, both of which also may be influenced by sex, can also contribute to different brain function. There is another factor, however, which Dr. Miller touched on that often gets overlooked, and this is that every neuron, glia, or other cell type within the brain carries either the complement of male or female chromosomes, but not both. Long ignored as a crucial factor, the importance of either sex genotype versus external influences is beginning to emerge. The role that sex plays in brain development and function is becoming more apparent as our research progresses. We've come a long way from when Dr. Arnold Bernthal monitored the behavior of roosters after removal of their testes in the 1800s, but more in-depth research is needed to continue our understanding of this field. We have some truly outstanding investigators here today to share how their research examines sex differences in brain function and behavior. I would like to invite our first presenter, Dr. Kyle Burghardt, to the podium. Dr. Burghardt is an assistant professor at Wayne State University, and the title of his presentation is The Influence of Metabolic Syndrome and Sex on the DNA Methylation in Schizophrenia. So please join me in welcoming Dr. Burghardt to the podium.
Thanks. All right, thank you so much for that introduction, and I'm really glad to be here to talk to you about our work where we're looking at essentially epigenetics of metabolic syndrome in schizophrenia. So first, I'd like to acknowledge my collaborators on this grant. First of all, Vicki Ellingrod, who is the PI on the R01 that we got the supplement for this grant. And then Jacqueline Goodrich and Dana Dolnoy, who are epigeneticists at the University of Michigan. They really helped us with the omics portion of our study. As I mentioned, this was supported by one of the clinical gender supplements, and we also used some of the core resources available at the University of Michigan. So today, in this short talk, I just want to give you an idea of what we're looking at in terms of DNA methylation and kind of stratifying that by sex and perhaps its potential role in schizophrenia. And then I'm going to give you some of our just top findings from our epigenome-wide association study that we conducted. So just to give a little bit of background in case you might not know, what is epigenetics? And there's a lot of different definitions out there. In terms of my talk, it's really just how the cell controls gene expression, at least one of the mechanisms of how it controls gene expression. And arguably one of the most studied forms is that of DNA methylation. There are other epigenetic modifications that occur in the human body, but today I'm going to talk about DNA methylation, which you can see is at the bottom here. And it occurs throughout the genome. It occurs most commonly where cytosines are next to a guanine, called a CpG dinucleotide. And then on a cytosine molecule that you can see here, it's where a methyl group is added to the 5th position of the cytosine. So in general, when you have methylation, it leads to gene repression, and then the opposite. But there are exceptions to that rule as well. We have the ability to study genome methylation at a global perspective using restriction enzyme approaches and transposable elements. And then we can parry down all the way to the site-specific methylation as well. And epigenetics has really garnered a lot of attention because it kind of bridges a gap where it can be stable in certain areas of the genome and passed on from generation to generation, but also seems like it can be influenced by environmental factors like diet, lifestyle, and medications. So the problem that we're looking at here in our work is metabolic syndrome and schizophrenia, which has been shown to be about 30% higher in schizophrenia compared to the general population. And down here at the bottom is a little snapshot from a meta-analysis which shows the rate is 32% higher in schizophrenia compared to the general population. And then furthermore, there seems to be some sex-specific effects where within schizophrenia itself, it's actually elevated in females compared to their male counterparts. And it could be occurring for many different reasons, but as a pharmacist, we're really, and Dr. Ellinrod's a pharmacist as well, we're really interested in looking at the treatment that they're on. And so the atypical antipsychotics are well known to cause acute metabolic side effects that include weight gain, dyslipidemia, and obesity, which makes up the metabolic syndrome. I should mention that the metabolic syndrome is a constellation of symptoms, metabolic dysregulation symptoms that include obesity, dyslipidemia, hypertension, and yep, I got those 3. And so this is really important to us because it's been shown that the metabolic syndrome leads to an increased risk of diabetes and cardiovascular disease, and the patients with psychotic illness, specifically schizophrenia and bipolar patients, who are the main receivers of atypical antipsychotic treatment, actually have anywhere, depending on the study that you look at, 10 to 20 years of premature mortality compared to the general population. So we're treating these patients with these really helpful drugs for the psychiatric symptoms, but we seem to be sacrificing some of their metabolic or physical health. And so in our lab, we're really interested in investigating the mechanisms by which this occurs. And the epigenetic hypothesis really started about 10 years ago with Dr. Ellinrod's work in the folate cycle. So this is a really simplified folate cycle on the left. And what she found was that patients that carry a reduced functioning form of this gene called MTHFR, or methylene tetrahydrofolate reductase, they actually have an increased risk of metabolic syndrome. And so when I came into her lab, one of the things that I found out was that one of the main endpoints to the folate cycle is actually that of producing methyl donors for various reactions in the cell, including DNA methylation. So I asked her, maybe there's some sort of differential methylation occurring that's underlying this metabolic syndrome risk in patients with schizophrenia treated with atypical antipsychotics. And so we published a preliminary study in 2012 where we found that when we looked at global methylation, so that snapshot of the genome's methylation, it was actually lower or hypomethylated in patients with metabolic syndrome, so schizophrenia patients with metabolic syndrome, and especially it was even lower in females with metabolic syndrome compared to the females without metabolic syndrome. And so we were able to use this preliminary data to get one of the gender supplements to really start to hone in on certain areas of the genome with an epigenome-wide association study. So for this study, we used the most common one is the Illumina 450KB chip. They've actually increased the size of this chip recently, but when we did this study, there was about 450,000 sites throughout the genome that's covered. And we utilized samples from Dr. Ellinrod's R01, which is a unique sample set in that she really deeply phenotypes these patients for both psychiatric and physical illness. For the epigenome-wide portion of the study, we matched our males and females on clinical and demographic characteristics. And then we used the accepted bioinformatics strategies to normalize our omics data. And then essentially what we were looking at is which CpG islands, which are heavily dense methylation regions in the genome, which are considered important for gene regulation, which of these CPG islands are most differentially methylated in males or females with or without metabolic syndrome. And then, due to the limitations of sample size and funding, we wanted to try to validate our top findings with the gold standard, site-specific pyrosequencing. So a little bit about the study population. They were all assessed for metabolic syndrome using the ATP3A criteria. They all had schizophrenia. And it's important to note that we used peripheral blood samples for this epigenetic study. That'll lead to certain conclusions that we can make from this data. All these patients were stable on their antipsychotics for 6 or more months, and we screened them out if they had a metabolic abnormality before starting their atypical antipsychotic. So here's the EWAS sample. As you can see, it was one array, so 96 samples, 49 males, 47 females. And we matched them fairly well in terms of age, average about 49. A half to two-thirds were Caucasian. They had a very high rate of metabolic syndrome in this group, so about 60%. There was a little bit of a difference in current smoking rates. However, this actually didn't reach statistical significance. And then we broke them down by antipsychotic use. And so we as pharmacists do this because the atypical antipsychotics actually cause metabolic side effects to differing degrees. So the olanzapine and clozapine cause metabolic side effects most often, so you have the highest risk with those. Putative, risperidone, and paliperidone are kind of middle of the risk. And then finally, aripiprazole and ziprasidone are low risk. So about a quarter on those high risks, and then the rest were distributed for middle and low risk. And here's the Manhattan plots from our epigenome-wide association study, where on the Y value you have p-values, Y-axis you have p-values, where as you go up on the Y-axis you have a more significant p-value. X-axis is chromosomes, 1 through 22, and each dot represents a CPG island. So as you go up, like this one here, was our most significant finding in males. But really what you're just looking here is visually comparing the epigenetic profile between the two groups, so male and female. And what you can see here, if you really were able to maybe apply a smoothing function to this, is that their epigenetic profiles are different. So let's just look at the top hits in both groups. In males, it was in this gene called CCDC8, and so they had a positive fold change. For our study, that meant males with metabolic syndrome had increased methylation at CCDC8 in that CPG island. So it was hypermethylated compared to males without metabolic syndrome. In females, this gene called MAP3K13 had a negative fold change, meaning that females with metabolic syndrome had hypomethylation or lower methylation compared to females without metabolic syndrome. And so what we did was, again, validate those top hits. So in an additional sample of 166 subjects, 100 males, 66 females, we tried to validate these top hits. And as you can see, again, they're pretty well matched in terms of age and race, and their smoking was better matched this time. And then the distribution of antipsychotics, atypical antipsychotics, was fairly similar to before. And this is a busy slide, but really what I want you to draw from here is that we looked at 4 sites with pyrosequencing in CCDC8 in the males, and then we looked at 4 sites with pyrosequencing in MAP3K13 in the females. And what we found was that we weren't able to validate our finding in males, so even in the crude or the adjusted for age, race, and smoking and antipsychotic type, they were not significantly correlated to metabolic syndrome. While in females, we actually were able to find 3 different sites just in the area that we chose to look at that were associated with metabolic syndrome. Again, where hypomethylation was associated with metabolic syndrome. So what are these genes? Just to kind of give you some food for thought. CCDC8, coil domain containing 8 gene, or protein phosphatase might give you a better idea of what its function is. It has several important functions within cells. And I think this post-translational modification of proteins is really important here. But what I found from the literature was that there is some sort of metabolic effect in this gene in terms of it has influenced insulin receptor alternative splicing. So insulin resistance is one of the defining features of metabolic syndrome. So there seems to be some, at least, support from the literature that this gene might be involved in metabolic processes. The MAP3K gene, mutagen activated protein, kinase 13. It is a serine threonine protein kinase, and it affects the JNK MAPK pathway. And again, these are just 2 studies that I found. There's more where they found differential methylation or expression in the endothelial cells of mothers with obesity, but also it's been identified as potentially a candidate gene for diabetes. So again, this kind of insulin resistance, diabetes theme that follows with this gene as well. So where are we going to go with this work? I think that because it was done in peripheral blood, it's hard to draw mechanistic conclusions, and the fact that it was cross-sectional. But I think there could be some biomarker utility here. I think in the future, we're looking to look at gene changes before and after treatment with an atypical antipsychotic, either within schizophrenia or maybe other paradigms as well. And then I think perhaps we need to consider, and in my group, we're looking more tissue-specific in nature. In my group, we're starting to take skeletal muscle samples from bipolar and schizophrenia patients and look at gene methylation changes before and after treatment with an atypical antipsychotic. And then here are my references. You'll be able to look at them if you download the slides. And then this weekend, I have to go give my 1-year-old a birthday, so I thought I would show you how excited she is. Be happy to take any questions.
Thank you very much for that. I'm not familiar with the EWAS thing that you did in the computer programs that are available, but I will say from our organization, we have done a lot with GWAS studies.
Okay.
And one of our former Birch scholars has developed a program that accounts for the sex chromosomes within these studies and also accounts for exon activation. That program is available free on the web. I don't know the address, but I can give you the contact information because I think, especially for the women, I noticed I couldn't figure out, I went too fast, I didn't think about the hormonal status of these women, but some of these genes associated with insulin regulation, also if there's interaction between the estrogen and some of those genes, the PPAR gamma and so on and so on. So I would like to offer you the opportunity to perhaps explore this other analysis tool for your data to see if you can pull out some additional genes that might be important in those women.
Yeah, that'd be fantastic. I think that as we keep pursuing the sex-specific differences, I think there is a big overlap in that insulin regulation and kind of the specificity maybe to females especially.
Hi, Tom Curry, Kentucky. Really interesting talk. So in terms of the female with a MAP3 kinase 13, so that's a signaling pathway. How is that related to some of the metabolic syndrome? Is there any tie between that and the metabolic syndrome in terms of the potential signaling pathways?
Can you repeat the last part of the question?
So is there any tie between the MAP kinase signaling in terms of phosphorylation, serine types of things in relationship to how metabolic syndrome might develop and or be expressed?
Yeah, I think that connection is mostly just with the insulin resistance portion of metabolic syndrome. So I think it's serine 3 kinase ability. It targets several proteins within the MAPK, JNK general pathway, and that influences insulin signaling pathways in that manner, if that answers your question. I think we have one more.
Carl, thank you. Can I ask a question?
Yeah.
So I was really interested also that you were trying to incorporate ethnic diversity in your samples. So you had Americans of European and African origin. I'm kind of wondering if you're introducing more variability there. And, you know, we don't know a lot about epigenetic differences in a trans-ethnic manner. But I think it's something to consider. And I don't know whether, you know, when you do your statistical analysis, whether you build in ethnicity as a confounder.
Yeah, so in both the EWAS and the validation, we included it as a covariate to adjust the model. So the EWAS uses linear regression as the model. Lima is what the package is called. And so we included that as a covariate. But I agree that I think there's a lot of work that needs to be done in epigenetics in general in terms of the race and ethnicity differences. In terms of our population, we recruit from southeastern Michigan. Ann Arbor is where we got these participants from, so it's really a reflection of Ann Arbor's demographics. Thank you.
Thank you again, Dr. Burkhardt, for a truly excellent talk. Our next speaker is Dr. Eugenia Trushina, an associate professor at the Mayo Clinic in Rochester, Minnesota. Dr. Trushina's talk is titled Sex-Specific Metabolic and Epigenetic Changes in Primary Fibroblasts from Patients with Alzheimer's Disease.
Thank you. Thank you very much for this great opportunity to talk about our work. The focus of the laboratory is actually on mitochondria. So we have a very robust way to look, starting from basic science applications, very basic molecular mechanisms, to look at what happens with mitochondria on the level of mitochondria dynamics, axonal trafficking, fission fusion, mitochondria function, under different conditions that include stress associated with neurodegenerative disorders, hypoxia, aging, and we're trying to spin this all the way through human translational program, through human samples, human tissue, because we believe that energy is at the bottom of all problems and processes that happen with us with life. And I think my major message today, and I hope that will be the home take message, that women and men respond to stress differently, and they respond to energetic stress in a different way, dipping in a very different sources of compensatory mechanisms, and that could underline the different progression of different disorders. It could underline the response to different therapies, and that basically tells us that all approaches probably will need to be tailored to different age, different sex, and different stage of the disease. So the focus of my talk today will be on Alzheimer's disease. And what do we know about Alzheimer's disease? Unfortunately, very little. So these tangles and plaques are the major hallmarks of Alzheimer's disorder, and the disease basically doesn't have any genetic component. So it's a sporadic disorder. That means that we only know a couple or handful of risk factors, including the ApoE4 allele, but it happens without any strong genetic component. So why and how? We do not know. Age is the greatest risk factor, and there is a huge sex component that I will talk about on the next slide. But what became increasingly recognized is that the failure of every single clinical trial conducted today focused on changes of the level of amyloid protein or the clearance of amyloid protein. They failed. Every single trial failed. What does it mean? That we do not really have a very good understanding of the mechanism. We do not understand early mechanism because, as Dr. Miller pointed out, the symptoms or the development of the disorder happens at least 20 years before the manifestation of clinical symptoms. So how does it happen? Why does it happen? And how can we monitor these early changes, which probably will give us the best way to diagnose the disease, to develop early treatments that could be more efficacious compared to these late trials in recent clinical trials. How can we do that? We do not really have these early diagnostic markers, and this is a big problem. So what do we know about Alzheimer's disease? That there is a very early perturbation in glucose metabolism that could be detected with the FDG PET, and this is the very first abnormality, so reduced glucose utilization in the brain, that is seen in patients with mild cognitive impairment, a prodromal stage of Alzheimer's disease. This reduced glucose utilization was actually directly linked to mitochondrial dysfunction on the level of changes in the components of oxidative phosphorylation that occurs in mitochondria, and that is directly linked to the memory loss. Sex-specific changes in Alzheimer's disease are well documented, but for a very long period of time, this prevalence of Alzheimer's disease in females was associated with the longer lifespan in females. But now it became clear that it's not true. So women are disproportionately affected on multiple levels, not only as patients, but also as caregivers. and biological mechanisms underlying the sex-specific changes are not well understood at all. Women who are ApoE4 carriers, similar with men, more predisposed to have Alzheimer's disease, but they respond very differently. So they have much more stronger aggressive symptoms, but they actually live longer. That seems that they could cope with this problems better than men. And basically, again, that suggests that there are differential mechanisms underlying Alzheimer's disease, and we have to create very specific, sex-specific cure, and men and women probably will not be treated in the same way. What are the gaps? So what is the biggest problem in the field right now? How are you going to study the disease? Where are you going to study the disease? So the failure of clinical trials brought up huge attention to animal models. To what extent preclinical animal models that we have to study Alzheimer's disease recapitulate human conditions? You cannot really get any brain cells from living patients. So you can study periphery. You can study blood, changes in CSF. CSF is very hard to get, and blood could inform only to certain ways. Dr. Miller again brought up a very important question, that there is a talk between brain and periphery. So could we use peripheral cells and detect changes that are associated with changes in the brain? And then you may have better model along with animal models, right? Because we cannot go without animal models to have longitudinal, nice systems to study manipulations and responses, understanding, of course, the limitations of these systems. But can we get some primary cells, peripheral cells, such as, for example, fibroblasts or lymphocytes, that we can get from the patients and study translational aspect of the disorder? So that's what we were interested in based on our preliminary data. So with this question, how periphery recapitulates changes in the brain, we did this study using samples from Mayo Clinic study on aging, and we have CSF and plasma from the same patients that either have mild cognitive impairment or Alzheimer's disease. So this study was published in 2013, and we applied systems biology approach, the newest omics called metabolomics. Why? Because our hypothesis was that these are metabolic changes associated with mitochondrial dysfunction, associated with abnormal glucose utilization. We should detect these changes on a systems biology level, and metabolomics could help. So metabolomics detects small metabolites, such as components of the TCA cycle, amino acids, lipids, a number of small metabolites that actually in the body exists in thousands of molecules. So it's a very robust method that doesn't really allow you to follow one particular pathway, but allows you to look at multiple interconnected pathways. So it's a powerful tool if it's applied properly. One of the interesting observations was, and you can appreciate that we found that in CSF, for example, the changes in the TCA cycle, so again, this is mitochondrial dysfunction, were the most pronounced. While in the plasma, it also was on top of the pyramid. In red, you can see the overlapping pathways, and most of them were associated with energy pathways. We did this, again, in MCI and in Alzheimer's patients, and our conclusion there was that, to our big surprise, changes in plasma recapitulate to 60% changes in the CSF. So it's very accurately represent changes in the brain, which was very promising. So if we can detect changes in metabolomics in the periphery, we can use blood for blood-based biomarkers for the disease onset and progression. And then the question became, can we use, for example, peripheral cells? But one of the things that I'm showing you right now is also our early metabolomic studies when we did metabolomics in the brain tissue of transgenic mouse model of Alzheimer's disease. So this is double transgenic mouse model that carries mutations in APP, amyloid precursor protein, and PS1. So we look at males and females, brain versus plasma. So first thing, which was very positive, that these mice do recapitulate early signatures of early energetic stress, similar to human samples. But we found that female mice are affected to the great extent than male mice. So I'm not going to talk about this further, but this is, I think, a very important connection showing that animal models should not be dismissed because they also could provide very informative information on disease progression and actually be used to test therapeutic strategies. Another application of animal models was very interesting. So the hypothesis became that changes seen in males and females could be this differential response to alternative utilization of alternative fuel to maintain brain function. So what does happen when the brain cannot any longer utilize glucose? There should be something else that brain can utilize to compensate for maintaining brain function. Usually, normally it could be amino acids, lipids, fatty acids, and ketone bodies. So ketone bodies could be produced either in liver and delivered through the bloodstream in the brain, or as we found in collaboration with Dr. Brinton, who actually a big proponent of these sex-specific differences in terms of alternative fuel utilization in postmortem in the brain of postmenopausal women. We did the experiments in animals, female brain tissue of mice that were postmenopausal, and we found that they also have abnormal glucose utilization, but instead of dipping in liver-produced ketones, they actually catabolize myelin in the brain that led to brain tissue, white tissue degeneration. And this is the common thing observed in female postmenopausal brain and also in people with Alzheimer's disease. So there is something different about how females dip into the potential compensation for this brain glucose utilization. And that could be the utilization of brain fats, not liver ketone bodies, which is very dangerous because that basically will lead to dementia and white matter degeneration. This is the study that was published very recently by the Metabolic Consortium, which was led by one of our collaborators, Dr. Rima Kadura-Duk. And this is a very important study because it's the largest metabolomic study applied to Alzheimer's disease. The study utilized blood samples collected within ADNI studies, and it's about 700 people. And what was found, that depending on the pathological markers of the disease, because these patients are very well characterized in terms of imaging data, in terms of cognitive data on cognitive tests. But what was amazing, that depending on what biomarkers of disease progression, of Alzheimer's disease progression they had, changes in metabolism were actually different. This is extremely important because that really supports the observation that you can longitudinally observe changes on metabolic level in blood associated with progression of disease in the brain. So here you can see the major pathways that were associated with each of the pathology markers. And it's interesting because these are all, again, these are compensatory mechanisms that could be engaged in alternative fuel utilization. So our hypothesis was that metabolic changes associated with mitochondrial dysfunction underlie early molecular mechanisms of Alzheimer's disease. And if we can find any association of metabolic and epigenetic changes, that could really, in fibroblasts, that could actually provide the disease model for translational research. And we could study molecular mechanisms because these are cells, and we can really manipulate multiple pathways in the cells, again, looking at the outcomes such as metabolic epigenetic changes. We can test therapeutic approaches that we actually have in the lab. We have multiple patents now on small molecule compounds that modulate mitochondrial function. I don't have time to talk about this, but this is very relevant to big translational program that we're trying to establish and develop. And we can advance the field of individualized medicine because, I become very convinced that this is an individualized treatment because we all will, hopefully not, but some of us may develop the disorder and we need to be assessed individually depending how we develop this condition. So what we did, we acquired fibroblasts and control and familial Alzheimer's disease fibroblasts. So all fibroblasts were from Cariell cell repository. Familial Alzheimer's disease fibroblasts had the mutation in presenilin 1. So this is one of 3 mutations that occur in about 1% of the population. And if the person has this mutation, the development of disease is given. Majority of people, as I said, have sporadic Alzheimer's disease called late onset Alzheimer's disease, so there is no any genetic mutations. And we had fibroblasts from people with multiple sclerosis as the irrelevant disorder to actually validate changes, how specific metabolic changes would be to Alzheimer's disease versus other neurodegenerative condition. So we have equal representation of males and females. They were all white, and you can see how close they were matched by age. This is our workflow. So we were very naive when we acquired all these cells. The plot was great. The goals were straight. The approach was straightforward. So 20, 10, 6, 36. And we only wanted to do metabolomics platforms based on our plasma and CSF data. So we wanted to look now at targeted metabolomics when we will specifically measure the levels of components of TCA cycle, acylcarnitines, because that's the fatty acid pathway of supplementation of alternative fuel, and amino acids. Then we want to do fluxomics, because now you can really, in living cells, in real time, look at the incorporation of your stable isotope tracers that will not give you just the time shot, snapshot, but will give you the dynamic of the work of TCA cycle. And we used 2 tracers, glucose tracer, glutamine tracer. And we also wanted to look at epigenetic changes, as the previous speaker described. Epigenetic changes could be very important, explaining the nature of sporadic disorder. And then we will do data analysis, and we are done. So we're still not done, so it's work in progress. And the very first question was, how are we going to do that? So with all this number of cells, how are you going to do that? If anybody here had any experience with analytical platforms, you know that ideally, you have to run all these samples the same day, collect them all the same day, process them within the same time, run them on the assay side by side in triplicates, and then analyze data, and then you will be very accurately comparing apples to apples. If you do samples 3 months ago, put them in the freezer, collected the rest today, and then subject them to the analysis, your first batch will be 3 months in the freezer, and you have no control over any decomposition of your sample. And what you're measuring 3 months ago may be different from today because of the sample collection and storage. So instead of being done in a month, we first actually underwent a very robust assay development. For people like you, if you will ever want to measure metabolomics in fibroblasts and you want to go with decent numbers for your statistics, how are you going to do that? So we're preparing this very elaborate and advanced analytical paper for Nature Methods, because I think it will be important for everybody. So the postdoc in the lab, Jordan Wilkins, who's a very talented postdoc and has a lot of patience, he did this enormous comparison of how you culture samples, how you collect samples, what type of even collecting cells, through trypsinization or simply scraping the cells, or through methanol fixation and extraction, or through evaporation of the sample. What gives you the best collection of metabolites for each analytical platform or for each targeted approach, including acylcarnitines, if you want to look at fatty acids, amino acids, if you want to look whether your cells use amino acids as an alternative fuel, or the function of TCA cycle. So he did all this, and then he also did comparison of the recovery of metabolites depending on storage, 48 hours, 2 weeks, 4 weeks. And based on his conclusions, we were able to really process all our samples for metabolomics. So the same thing we're doing right now with tracers, because even if we did some of this tracer work, there was a lot of variability, and we need to look at this carefully, again, developing the assay for multiple cells. But what we found that indeed fibroblasts from males and females do recapitulate abnormal glucose utilization. So this is good. Then we looked, and this is busy, but don't worry, all you need to do is to look at males versus females, males versus females. When we look at metabolic changes, you can appreciate that metabolic changes in males are much more pronounced than in females, and we saw it with both tracers and stable isotope tracers and targeted metabolomics. We look at what does it mean that these metabolic changes are different. How does that reflect to the ATP levels in the same fibroblasts and mitochondria DNA copy numbers? So we found that male fibroblasts have high DNA copy numbers, so there is more mitochondria, but they have less ATP production, while females are unaffected. So something is very different in males, and again, kind of telling you about our conclusions, we think that males cannot compensate as efficiently as females. We use these metabolic biomarkers in fibroblasts to test whether we can use them for diagnostic. And we utilize this collaborative laboratory integrated reports called CLIR, developed at the Mayo Clinic by Dr. Rinaldo and his colleagues. And I can talk about, if there is any interest in details about that. But this is a post-analytical tool specifically designed for metabolomics data. It's 47 countries contribute to this database at the Mayo Clinic. So you subject your data, and the program allows you to look not only at the actual changes in metabolites, but also ratios. And the program will run through all your data and basically establish the best separation of the groups. And you can see here that we have control subjects, and we have subjects with Alzheimer's disease. And then when we put one of our unknown sample, but we knew it was Alzheimer's, the system allowed to put it very accurately in the disease group. So metabolic changes in fibroblasts also have the ability to be used for diagnosis of the disease. So this is a comparison to multiple sclerosis, and changes are completely different. But what was interesting that you also see that changes in multiple sclerosis patients are also different between males and females. The next question was, do we see epigenetic changes that were detected in the brain tissue, postmortem brain tissue of Alzheimer's patients? Why is it important? Because it became very clear that mitochondria, changes in mitochondria, could actually affect directly DNA methylation, acetylation, and changes in, for example, ketoglutarate that we detected, changes in amino acids, ATP, could directly affect methylation. So we looked at the methylation patterns in this fibroblast from males and females, and we totally detected large number of differentially methylated regions, and among them, the majority of these changes were in females. Yes, I'm almost done. So we compared what these changes are in terms of brain tissue, and that's what we identify in fibroblasts, which is similar to previously reported changes in the brain. And these are the particular pathways that, again, are different between males and females. And it's the last slide. So when we plotted that using pathway analysis, these are particular markers of Alzheimer's disease, and changes in females are very directly related to these markers, while changes in males are a little bit delayed. So again, demonstrating that there is substantial differences between male and female on the systems biology approach level. So the conclusion is that fibroblasts could be used to study the disease, to diagnose the disease, and could be a nice model to test therapeutic approaches. So with this, I just want to thank all my collaborators. So it primarily was the work of Jordan Wilkins in the lab, but we, of course, have a big group of people on all levels, and without the sponsors and support from the NIH and common funds, this work will not be possible. We learned a lot. We learned that this is a very tedious and very expensive task, but I think it's very important, and we're looking forward to hear about any possible collaborations or grant opportunities to move this forward. Thank you.
Thank you, Dr. Trishina. I think we'll maybe move some questions to the break if you want to reach out. So our next talk is from Dr. Emmerin Mayer, who's a professor at the University of California in Los Angeles. And Dr. Mayer's talk is entitled Sex Differences in the Brain, Gut, and Interactions.
I'd like to thank the organizers for the opportunity to talk about our work here, and I also want to acknowledge a couple of people at the beginning. I'll do it again at the end. Annie Gupta and Lisa Kilpatrick, who have who actually joined UCLA in our center because our score, the early phases of the score, and have sort of become the main driving forces for sex difference research, both in brain-gut interactions. I'm following the sort of outline that was suggested to us. So the importance of sex differences in the field of brain-gut microbiome interactions, shortcomings, and also present some of the latest data that have come out of our UCLA score. Brain-gut interactions weren't such a hugely interesting thing to most people until the microbiome came on the scene. But since then, the last 5 years, it's kind of exploded. The interest has exploded both in the media and in funding agencies and scientific circles. A simple way to display these interactions, I could obviously give a talk about this here, is that we have several major players in this bidirectional communication, the gut microbiome, what's been called the gut connectome, a series of cells, immune cells, nervous cells, and endocrine cells that interact with each other and with the gut microbes that are closely adjacent. And the central nervous system, which is the brain connectome, multiple networks that are interconnected and generate the output. So what's important is the interactions go from the brain to these peripheral sites, to the gut, and both of these sites can send signals to the brain to modify function, structure, and development. Most of that, the role of the microbes, has been demonstrated in animal models, so we have to be careful how much of that actually applies to human disease. We got a supplement for the score to look at the microbiome. It was not in our original application, and unfortunately I will not be able to show you these data because they're still being analyzed. So as I said, the brain-gut microbiome axis has become quite popular and has been implicated in a variety of human chronic diseases, chronic visceral pain, functional gastrointestinal disorders, IBS, and obesity and ingestive behavior. I'll just show you a few things, or highlight a few things, both in terms of the efficiencies, in terms of sex differences, as well as some of our initial data to look at that. So why is the study of sex differences important for these brain-gut microbiome disorders? So first of all, we've known we're now in our third cycle of the score, that the brain is quite important, and there's often sex differences that are stronger than disease differences. So when we look at the healthy control group, we see larger differences than if you compare the control group with for example, with IBS patients. And also, as we know, and I'll show you some of these numbers, clearly plays a role in obesity. In both of those areas, these examples of brain-gut microbiome interactions, they're both sex differences and gender differences. So the way that affected patients interact with the healthcare system are clearly also driven by gender. So it's not just a sex difference area. So we've known for quite some time that a significant number of chronic pain conditions including the ones that we're interested in, this interstitial cystitis, or as it's now called UCPPS, and irritable bowel syndrome. So the major examples for chronic visceral pain. But we know that in all these chronic pain conditions, there's a greater prevalence in female populations. This becomes even higher if you look at the affected patients that seek healthcare. So these from population-based studies. But if you actually look at the clinics, the difference gets much higher, several-fold higher. So this is also a population-based study for IBS, and you see that the increased prevalence in women is not as high as was initially proposed from clinic studies, where it's like 10 to 1. So it's about 1.5 to 2, depending on different areas of the world and different kinds of studies. This has also varied with a real problem with all these functional syndrome studies with the symptom, since we only have symptom criteria. Every time the symptom criteria are being changed, the prevalence rates and the relative ratio between males and females varies. So clearly chronic visceral pain, an important area for studying sex differences. Obesity, equally important. So we all know there's an epidemic going on in this country and around the world. If you look at, just break it down into men and women that are obese, somewhat higher in women, not dramatic. But then if you look at, break it down into minorities and women, we see that amongst African Americans and Mexican Americans, females have a much higher proportion of obesity. And the area that we're interested in is what leads, one factor that leads to obesity, ingestive behavior, what is hedonic eating. So shortcomings of studying sex-related differences in brain-gut microbiome science. Here's an example that Annie Gupta and Lisa Kilpatrick put together in a review article, looking at 412 manuscripts that identified chronic pain neuroimaging studies. If you look at first, she had a functional chronic pain. So you can see that out of this large number of studies, only 15 manuscripts identified, could be identified that looked at sex-related differences, despite the much higher prevalence of chronic pain and chronic visceral pain amongst women. And if you break it down into the different pain conditions, you see that these 50 manuscripts make up a very small point, and the fact that it's highest in IBS is related to the SCORE funding, because I think we're probably the only group that looks at sex differences in brain function in syndromes. Another way of looking at this complex slide, so this co-citation network analysis of chronic pain neuroimaging studies with a focus on the treatment by sex. So what you see in green are studies that looked at or that reported sex differences. In red, they were female-specific, blue, male-specific, white, mixed sex. The size of each of these circles represents the total number of citations by December 2015. And I just want to point out this again, what I showed in the previous one. Even though fibromyalgia and migraine much more common in women, there's no green in here, so basically no sex difference studies. The only significant number is really in the IBS domain. So there's clearly a major shortcoming in looking at this at this question: how is the brain different in these prevalent chronic pain conditions? If you look at obesity, and again looking at the brain imaging studies, if you look at the green portion of, so obese only, lean only, and comparing obese versus lean. Again, you see the same phenomenon, a very small number of studies that have reported sex-related differences. I mean, remembering previous presentations by Janine, similar things apply probably to most human diseases, but it's kind of striking to us working in this particular area. So a few examples of studies that we have been doing. We have been fortunate to be in the third cycle of the SCORE funding, and have really over those 15 years published a lot of papers. Every single one had the sex difference aspect in it. Just give you sort of a very brief overview of what kind of topics we looked at. We looked at early adverse life events, and in this case, resting state neural networks. Similar study, sex difference in the influence of body mass index on the architecture of brain networks. Another series of papers. This was a review article. But we didn't just look at the brain, but also the outputs, autonomic nervous system outputs, and HPA axis responses. So the summary from our brain imaging studies, which are actually turned out to be comparable to the few studies that have been done in other chronic pain conditions. Significant sex differences exist in the altered morphology, connectivity, and response to pain in patients with chronic pain conditions. And these studies so far indicate fairly consistent, more prominent primary sensory motor, structural, and functional alterations in female patients compared to male patients. Difference in nature and degree of insular alterations, greater insular reactivity in male patients in studies that used stimuli, either stress or pain. And inconsistent differences. So our initial hypothesis was that the main difference is maybe in the emotional arousal reactivity, but that turned out to be not the case in general. It depends on the kind of emotional stimulus to give. If it's an anger stimulus where males have a greater response as opposed to a fear or anxiety stimulus where women have a greater response. So this really did not come out. The most consistent finding, surprisingly, is really the sensory motor network in the brain that makes up the difference. So one phenomenon of these chronic visceral pain conditions is that most of the time they overlap with other chronic pain conditions, which are all more common in women. And one of the biggest risk factors to have more than one system affected is, again, female sex. So if you're a woman and have IBS, you're much more likely to have fibromyalgia or urinary symptoms or vulvodynia than if you're male. So that to us points that something, we've sort of hypothesized this early, it's really been confirmed by other groups, Dan Claw in Michigan, for example, that there's some process of central sensory augmentation together with what I showed you earlier, greater propensity of women and a greater abnormality in the female sensory motor cortex. So this could be one of the factors that mediates this. So from one of our studies in the urological pains field, interstitial cystitis, chronic prostatitis, just want to illustrate this last point about this comorbidity and this widespread pain. So Jason Koch, who did this analysis, he looked at individuals that on the body map indicated where they experienced their pain, either in the suprapubic region, which would be localized, or extending into the abdomen, or extending really to the whole body. So widespread chronic pain is obviously related to these overlapping chronic pain conditions, even though these patients may not qualify for a diagnosis of a GI problem or chronic headaches, but they clearly indicate that they experience symptoms there as well. The reason I show this is because the abnormality is consistent with what I told in the last few slides, that a structural abnormality in the sensory motor region of the brain, gray matter difference, and this structural abnormality increases from the patients that only had the localized pain to the widespread pain. In addition, so the resting state activity of the brain and the connectivity, this resting state network, show that there's an increased connectivity between the salience network, so the part of the brain that determines how threatening or bothersome a stimulus or a symptom is, with this same sensory motor cortex, again in a dose-dependent fashion, localized less connectivity than in widespread pain, increased connectivity. Lisa Kilpatrick did a recent analysis sort of studying further probing into this question, what is abnormal in the sensory motor cortex? And until recently, the only way you could actually look at a technique, an analysis technique, connectivity gradient analysis, that allowed her to look at each part of the sensory cortex, the postcentral cortex, how closely it's connected to the insular cortex, the different regions of the insular cortex. Again, looking at that same sensory network that I showed you earlier. And what you see, it's interesting, the sex difference is, so the color codes brain regions that have similar connectivity between insula and sensory cortex. And you see that as a much slower gradient in women from the red area, which is a less or weaker, a weaker connectivity to the insula, to these higher connected regions, as opposed to males that have a very different gradient pattern of how these are connected. So we now know it's not only structurally or functionally in terms of the oscillations, but it's also in terms of how the anatomical regions are connected by fiber tracts. There is this sex difference. It's all confirming something that we never really hypothesized in the beginning of our studies, that this would be sort of the major part of the brain that plays a role. Sex-related differences in the reward network in obesity. I'm going to go over this quickly. So these data show you from a graph theory analysis of connectivity of different regions of the central reward system with other brain regions in individuals with high BMI and normal BMI. So as you can see, there's a small difference if you just look at this without looking at the sex difference, but it becomes quite different, the connectivity that you see in males with a high BMI compared to females. So in obesity, so the reward system clearly plays a major role in driving ingestive behavior. The connectivity of that region with other brain regions is quite different in obesity, depending if you're male or a female. And yeah, so I'm going to come to a conclusion in just a minute. So you've seen a few of these graphs, these tripartite association networks, where you put several sets of data into a combined analysis that looks at the associations between these different variables. In this case, we looked at matrices from several brain networks, as well as shown here in green, results from questionnaires that, for example, the Yale Food Questionnaire, the SF-12, looking at symptoms. And so I just want to point out that, first of all, there's a difference between high BMI and normal BMI, how different brain parameters are correlated with these ratings of health. Just give one example here. This is the reward region in normal BMI correlated, very different with the executive control association in the high BMI subjects. But most strikingly, if you look at, if you break it down into different sexes, you see a very different pattern of negative health outcomes and hedonic eating in women with a high BMI are compared to women with a normal BMI. So I don't have time to get into this exactly, but you can imagine that this gives you an indication how different mechanisms produce very similar symptoms or behavioral traits, such as overeating in males and females. So to summarize, hopefully I gave you some indication that brain-gut microbiome interactions play an important role in the regulation of food intake and in chronic visceral pain conditions. Epidemiological studies demonstrate that women are more likely to be affected by these conditions than men, but only a very small minority of brain imaging studies have addressed this question. And our studies demonstrate significant sex-related differences in brain architecture and function, and in the relationship of these brain findings with behavioral and clinical measure. Like in many other conditions, I think what this shows is that different mechanisms in the brain can produce similar behavioral or clinical outcomes. That also implies that treatment approaches, that's also what we're most interested in, both pharmacologically but also behaviorally, for example, cognitive behavioral therapy, are probably targeting different parts of the brain, and men and women will respond differentially to those interventions. So group of people that contributed are key players in the score. Highlighted those, Amy Gupta, Lisa Kilpatrick, and Claudia San Miguel contributed to some of the data that I showed you. And sorry for going over time. Thank you.
Thank you very much, Dr. Mayer, for the excellent presentation. Our next speaker is Dr. Margaret McCarthy, a professor from the University of Maryland, and her talk is titled Incorporating Sex as a Biological Variable in Understanding Brain Development.
Okay, thank you very much. Just moving us right along here. Brain is complicated, so that's why it's challenging to study. It's the earliest organ to form and the last to mature, and this is just showing in a primate brain from a newborn to 2 years old in the cortex, and this just barely captures the complexity of what's going on over development. In the human, the prefrontal cortex is not considered mature until you're a 26 to 28-year-old adult. This means we have an enormous amount of time during which things can impact the brain differently in males and females. In fact, that's what the brain is designed to do. It is designed to incorporate experience into its structure and function, and we all know that the experiences of boys and girls and men and women are profoundly different. And so this is one of the great challenges that we have in studying the brain. We also know that all of the neurological and neuropsychiatric disorders that you are capable of suffering from have enormous gender biases. As Virginia told us this morning, I challenge you to name any that are not different in some way between males and females. This is divided into female biased on the left and male biased on the right. The size of the wedge is sort of the magnitude of the sex difference. But what's not immediately apparent here is actually the profound difference in the onset and the diagnosis of these disorders. All of those male biased disorders are disorders of development. All of those females disorders appear peripubertally or post-puberty, almost as if there's a temperature inversion across the lifespan, so that males are highly vulnerable early and females are highly vulnerable late. So brain sex differences are multifactorial, and they're determined by your genes, your environment, and your hormones, as we've all been talking about. And this begins with your sex chromosome complement, which is going to determine whether or not you have an ovary or a testis, and your testis is going to make testosterone, aromatized estradiol. And what we know is, though, that female brain is actually bipotential, and that if we treat it, just to go back, if we treat the female with testosterone or estradiol, we can convert it into male, whether we do it pharmacologically or whether it occurs genetically or some sort of natural experiment. And I think of this as that this is because Mother Nature wants brain sex to match chromosome sex. She doesn't really give a whit about your gonadal sex. No, I said it backwards. Oh my God. She wants gonadal sex to match brain sex. She doesn't give a darn about your chromosome sex, right? So it's all making sure that your reproductive physiology and your brain physiology and behavior are in sync. So the problem is that the timing of these influences, your hormones, your genes, and your environment and experience are not the same. So steroid hormones, which are, I would argue, the most profound important in terms of brain sex differences, they diverge during a perinatal developmental critical period for sexual differentiation of the brain. They're entirely absent during early postnatal life and childhood, and then they diverge again at puberty and remain so for the rest of your life. And that's sort of diagrammed here with the prenatal surge of testosterone in males. Then we have this period called the juvenile hiatus when there's no hormones, and then adult males at puberty where you get this testosterone, and it pretty much it wanes a little bit here, but it's hardly andropause, as the pharmaceutical companies would like us to believe. Females, though, they're going to have a period of cycling. They're going to get pregnant. They're going to lactate. They're going to cycle again. They're going to get pregnant. They're going to lactate. They're going to cycle again, and then the bottom drops out and they go through menopause. So if you're comparing males and females, say at birth, well, they are intrauterine. They're profoundly different in their hormone profile. Compare them at birth. They're profoundly different in their hormone profile. Compare them at puberty. Actually, females go through puberty 2 to 3 years earlier in human. So if you compare pubertal males and females, you're actually comparing a brain that is of a completely different age. Adulthood, profoundly different, even in older adults. So we never have a time other than a brief period of childhood when the hormonal profile is comparable in males and females. So this is sort of the challenge of early life programming, which is one is hormones, which is very profound. Then layer on top of that prenatal or postnatal stress, maternal immune activation, hypoxic ischemic insults, which we'll hear about later from Anna Penn, chemical insults, etc., are all actually acting on a brain that by definition is different between males and females. And a further challenge is that the brain doesn't heal like other structures. It just repurposes, and the capacity to do so is going to decrease with age. When we do think about sex differences in the human brain, environment and experience are continuous in their influence, and they're impossible to control for. They are probably also profoundly important in ways that we still don't understand, beginning even in utero. When we name the child, we paint the nursery a particular color, we pick out the clothing, etc. So if we're going to really get to biological basis of sex differences, we use our animal models. And my animal model has been the increasingly exotic laboratory rat, which is not the laboratory mouse. So if I think about what are the sex differences in the brain, this is my version of everything that happens in the brain. We start with neurulation. Well, even the formation of the neural tube has been shown to be different in an XX versus an XY fetus. Neurogenesis, glial genesis, migration, dendritic growth and differentiation, myelination, synaptogenesis, synaptopruning, apoptosis. Every single one of these endpoints, I could sit here and tell you a story about how it's different in male and female brains. So it's similar with all the diseases. I defy you to find a brain process that is not different in males and females. Each of those occurs in different regions at different times. This is the further challenge of the brain, and every region functions by its own rules, both in what it does in terms of behavior and physiology, and also how it is differentiated by sex. And yet these regions are all talking to each other, right? We're always talking about the connectome. So even what we learn in one region could easily be influenced by another region connecting to it. And then there is that darn cell type issue, right? The brain is not like the liver where we have hepatocytes and hepatocytes. In the brain, every cell is different from every other cell. And this is just all the different types, right? We've got the vascular system, as Virginia was talking about. We've got myelinated neurons, unmyelinated neurons. We've got astrocytes. We've got microglia. And then we do have some neurons in there, too. Those neurons themselves are enormously complex and different in every brain region on all sorts of different sort of phenotypes. So where do we begin to look for sex differences? Well, first, before we start to talk about sex differences, it's always worth reminding everybody not all sex differences are created equal. We use it as a very unitary term, but it is not a unitary phenomenon. There are sex dimorphisms. There are very few of them. They're the exception. It's the most overused term in the study of sex differences is the term dimorphism, which means two forms. Very rarely do we have two forms in men versus women. What we mostly have is sex differences, which are things that vary along a continuum, where males and females have more or less of something. We also have this increasingly emerging, fascinating phenomenon of convergence, divergence, latencies, and compensations, where the same endpoint we just heard about from Emmer and Mayer is you get the same endpoint, but you get there in different ways. You might have at baseline, males and females are the same, and in response to an insult, they fly apart. You have some where you're compensating for your reproductive physiology biology to get to the same endpoint. And so we have to be very creative about how we're thinking about sex differences. One of the most brilliant things about the SABV policy is to not dictate how you're going to study sex differences, so that the scientists are creative about thinking about what is it that's really going on, and a lot of action happening in this space. And then lastly, we have some sex differences that are just a matter of frequency. It's the same in males and females, but it just occurs more in one sex versus the other. So in my laboratory, we're looking very much at cellular and molecular sex differences, and we find that we have to really drill down deeply into particular endpoints to look at the sex difference, and we have to focus very carefully on one region at a time and one endpoint at a time. So we've done a lot of work on the preoptic area because of this phenomenon, which is that the dendritic spine density on the male preoptic neuron is twice that of the female neuron. And as you all know, these little spines here are the sites of excitatory synapses. And so we've been trying to figure out mechanistically how this occurred. And we found that developmentally testosterone aromatized estradiol binds to the estrogen receptor, goes to the DNA, and transcribes the cyclooxygenase enzyme, which makes prostaglandin E2, which acts on neighboring astrocytes, causing calcium-dependent glutamate release to bind to AMPA receptors and cause the formation and stabilization of these dendritic spines. So it's a cell-to-cell communication, actually involving a prostaglandin, not a neurotransmitter. Along the way, we found that we actually needed another cell type. So handily, we found microglia because we knew that we had another source of prostaglandins. And microglia are, now we know, the brain's immune system. They actually originate outside the brain in the embryonic yolk sac. They migrate in. And then they do this fantastic thing that they take on all these different shapes, which gives us an insight into their mood. So this amoeboid microglia is very, very worked up and is making lots of prostaglandins and cytokines, etc. This is a ramified microglia, which is just surveying and checking on everybody else's business and seeing how everybody is doing. And if we just, and microglia are really cool because they get into the brain before everything else important happens. So this is conception, embryonic day 15. They actually start migrating into the brain around embryonic day 9. And they're there and ready when oligodendrogenesis, astrogenesis, neurogenesis, angiogenesis, synaptogenesis, pruning, etc. are all going to happen. So they're there ready to modulate all of that. This is what those microglia look like in a male POA versus a female. You can see hers are all nice, filigreed, lacy, ramified, very busybody microglia. His are all worked up and blebby and angry, and they're making lots and lots of prostaglandins and cytokines. And as you can see, the whole population is completely different. Then along the way, we needed a fourth cell type, and we found mast cells. It was the result of an RNA-seq. Mast cells are the only other sort of naturally occurring immunocompetent cells in the brain. They come from a different origin. They come from the bone marrow. And as you can see, they are secretory machines, right? And they also are very motile. And to borrow Virginia's saying, they are the 911 responders whenever there's an injury in the brain. So we went around and counted them in the POA. They look like schmutz on your slide, these little bits of blue, but that's actually just a mast cell stained with toluidine blue. Here's how you count mast cells. 1, 2, 3, 4, 5, 6. We're done. So we actually count them in the tens. There are tens of mast cells in the brain. And you might say, well, that's pretty weird. It is, in fact, it's really fascinating. They are a very rare event, right? Yet they are profoundly powerful. This is a detailed characterization. The most we see is about 1 week after life in the males, and you can see they have many, many more than the females. This is the period during which the brain is differentiating in males versus females. Then they go down to almost nothing, and they pop back up again in adulthood, but are no longer different in males versus females. And what we found is that the mast cells are actually secreting histamine, which is, and unfortunately this is a video, but we couldn't get it to work, which is causing the microglia to make more prostaglandins, which is activating these astrocytes, which is causing the formation of these dendritic spines. So we now know we have 4 cell types involved in this synaptogenic process in the POA, which we've also tied functionally to behavior. And 2 of those cell types are non-neuronal in origin. Now, we also have been doing a deep dive in another brain region, the amygdala, because it's important to this juvenile social play behavior that occurs during that time when there's no hormonal difference in males and females. So any hormonal influence that there is occurred perinatally when they were born during the sex differentiation process. And we're again focusing on these microglia because we found along the way that they had these funny cup-like things, which is what a new graduate student in the lab said, what are those funny cup-like things? And the postdoc said, I didn't know what they were, so I just ignored them. I was like, oh my God, are you kidding? Look at these things. These are phagocytic cups, right? So this is what we found is that these microglia are doing is that they are in the ramified state, and then they shoot out an arm and they grab a cell and they pull it back into their body and they digest it. And there's a lot of this going on in this very early brain. Here's another example. You can see here is the microglia's cell body, and here I call this our Baltimore lacrosse microglia. This is our Mickey Mouse microglia. Here's a double D cup microglia. You can see they're very, very hungry. Right? So when we quantify these in males versus females, here we are beginning at birth. Here the male, this is just the number of microglia that are phagocytic. And this is an event that takes about 2 hours. We're taking a snapshot in time, right? And we see that the males have significantly more microglia that are phagocytic compared to the females for the first 4 days of life, and then the sex difference drops out. Here's if we look at the percentage of them, up to 30% of those microglia are phagocytic at any moment in time in males versus females for those first 4 days of life. Well, what are they eating? When Beth Stevens came and visited us and we showed her images of these, she said, holy cow. Of course, Beth Stevens is famous for microglia nibbling. She said, these microglia are going to town. They are absolutely gorging, as you can see this one here. Well, what they are eating is other cells, but what types of other cells? Well, PCNA is a marker of newborn cells, and here's a microglia with PCNA in its cup, so it's just engulfed a newborn cell, and it itself was only born recently. PCNA means the cell was born within the last 48 hours. They are proliferating like crazy, about 40% proliferation rate, and they're born hungry. Here's another image of using PCNA of a microglia with this huge phagocytic cup as our imaging has gotten better and better. So the question we have asked is this phagoptosis? Phagoptosis is a combination of phagocytosis and apoptosis. So it is the engulfment of living cells. We have found that about 60% of our cups are filled with newborn cells, and males have fewer newborn cells than females. And so we can directly correlate this phagocytic consumption to the reduction of new cells in males. And you're going to hear more about that when my postdoc gives a talk a little bit later this morning. So when we think of all of the things that are going on in the brain that are different in males and females, we have to add one more, which is murder. He was just a boy. So in my last minute, I'm going to do two sort of conceptual thoughts about sex differences in the brain and the challenges, because sometimes I could sit here and tell you 10 more stories about how the brain is different in males and females, and I could give you all sorts of cool mechanisms. We have the SDN or the POA, the most famous sex difference in the brain, the most studied as well. It involves caspase-3, calcium regulation. There's also the AVPV, which is NRF2 dependent. The hippocampus, we have a sex difference in neurogenesis involving depolarizing GABA, and I just told you about the amygdala, and I didn't tell you about the endocannabinoids. Catherine Argue will tell you about that later. Prostaglandins and microglia and the POA and the nearby arcuate, we have a completely different story involving GABA and astrocytes. And these are just a few of the many, many examples. But what that means is that there's no such thing as this. There's no such thing as a unitary male or female brain, because it's simply biologically impossible to have a different cellular mechanism in every single brain region and have it be unitary and all happen the same. When we have allelic variation, we have differences in timing and insults and hormonal exposure and environments, etc. So instead, what we're dealing with is a mosaic brain on a very biological, fundamental biological level. But so we can have a range, right, of females are going to have some regions that are masculinized, males are going to have some regions that are feminized, and to varying degrees all throughout. So, you know, it's a wonderful rainbow coalition. But a mosaic is not a blend, and that's a really important difference. What do I mean that by a mosaic is not a blend? Well, a blend would mean that we were all just a continuum of male and femaleness. But after 25, 30 years of studying sex differences, I have noticed a consistent pattern over and over and over again, which is when I do anatomical endpoints, not behavioral, anatomical endpoints, I find a sex difference that is always about twice in magnitude. Sometimes 1.5, sometimes 3, but always on average around twice, never 10 times, never 30 times, right? I think sometimes females higher than males, etc. If I give females hormone to make them male-like, they go right up to the male level. They never zoom past. And if I give males more hormone, I can't make a super male. I can't super masculinize a male. If anything, they usually get worse. They usually become dysmasculinized. I also noticed that the variances are small and highly consistent. So it's as if something is suppressing the variability within each sex. But something else is pushing the sexes apart, because they're consistently always about twice as apart. And what this is reminiscent of, as we were just hearing about epigenetics, it's Waddington's, one of his original concepts was self-fate, right? And that was via canalization. And canalization is a real biological phenomenon in which your fate is restricted because you went down one canal versus the other. And I propose that sex differences in the brain are in fact canalized, so that females go down one path and males go down another, and while they might vary within their path, they cannot cross over to the other side easily because there is a major barrier, but they also can't get too far out of their own canal because Mother Nature doesn't want males and females getting too far apart, because we all know that would be a bad thing. What are the agents of canalization? Well, heat shock proteins. What do heat shock proteins bind to? Steroid receptors. There's also microRNAs. There's epigenetics. There's environment and experience. You know, put down that doll, young man. I'll give you something to cry about. That's canalization, right? So and I think that's an important concept to keep in mind, and we are biologically testing this right now as well. Thanks to the funding agencies, particularly the Office of Research on Women's Health, I will be using my supplement to look at epigenetic changes in those microglia, and to all of the people in the lab who actually did the work. And I finished on time.
So that wraps up session one. Just like to take a quick second once again to thank all of our speakers. Those are really some great talks, so thank you guys very much. We will now have a break till 11:15, and after which we'll begin our second session of the day, which is how sex impacts our interaction with external influences.